Hello, welcome back to my blog. Today I'm going to be doing an in-depth blog on E. coli. So, Escherichia coli, or E. coli, is defined as a gram-negative, rod-shaped, facultative, or anaerobic bacterium that consists of hundreds of different serotypes and is highly versatile. These serotypes range from strains that are harmless and play a vital role in maintaining intestinal function, to strains that contain pathogenic properties that infect the human body and cause distinct signs and symptoms. Pathogenic Escherichia coli strains are capable of causing three common clinical diseases that include sepsis/meningitis, urinary tract infection, and enteric diarrhea disease. The pathogenic E. coli is known to be highly diverse, adaptable, and contain various virulence factors that affect a wide range of cellular processes. Therefore, pathogenic Escherichia coli is studied extensively in food, mammals, and the environment. Outbreaks of diseases associated with E. coli are common in developed and developing countries, which pose a major public health concern. Commensal isolate E. coli HS commonly inhabits the normal microflora of the human and animal gastrointestinal. E. coli HS is a bacterium that can be readily grown in a laboratory setting and has genetics that are easily manipulated. This serotype of E. coli HS is O9 and contains many synthetic sequences with other E. coli genomes. E. coli HS exhibits 94 genes that are unique to its serotype, while 64 of these genes do not demonstrate functional annotation. Of the unique genes that exhibit functional annotation are associated with the production of the serogroup O9 lipopolysaccharide. So E. coli can live on a wide variety of substrates and uses mixed acid fermentation in anaerobic conditions, producing lactate, succinate, ethanol, acetate, and carbon dioxide. Since many pathways in mixed acid fermentation produce hydrogen gas, these pathways require the levels of hydrogen to be low, as is the case when E. coli lives together in hydrogen-consuming organisms, such as methanogens or sulfate-reducing bacteria. In addition, E. coli's metabolism can be rewired to solely use CO2 as the source of carbon for biomass production. When growing in the presence of a mixture of sugars, bacteria will also often consume the sugars sequentially through a process known as catabolite repression. By repressing the expression of the genes involved in metabolizing the less preferred sugars, cells will usually first consume the sugar by yielding the highest growth rate, followed by the sugar yielding the next highest growth rate, and so on. In doing so, the cells ensure that their limited metabolic resources are being well used to maximize the rate of growth. A well-used example of this with E. coli involves the growth of bacterium on glucose and lactose, where E. coli will consume glucose before lactose. E. coli grows in a variety of different laboratory media, such as lysogeny broth, or any medium that contains glucose, ammonium phosphate monobasic, sodium chloride, magnesium sulfate, potassium phosphate, dibasic, and water. Growth can be driven by aerobic or anaerobic respiration, meaning with or without oxygen, using a large variety of redox pairs, including the oxidation of pyruvic acid, formic acid, hydrogen, and amino acids, and the reduction of substrates such as oxygen, nitrate, fumarate, dimethyl sulfoxide, and trimethyl N-oxide. The ability to continue growing in the absence of oxygen is an advantage to bacteria because their development is usually largely based on a water-dependent environment.